Hello, everyone, and thank you for joining this session on industry perspectives of the IHCC. I'm Scott Sunseth, the director of the IHCC, and I'm joined this morning by Cindy Lawley, who is the Global Director of Population Health at Olink Proteomics, Peter Wirtz, the Scientific Director and Founder of Nightingale Health, and if you heard about some of our metabolic projects yesterday, Nightingale Health was one of the collaborators on that, and Heike Runes, who's the Head of Human Genetics at Biogen. Thank you all for joining me this morning. We're going to go do a quick round robin and let each of the panelists go and do a 5-minute intro of their organization. Cindy, we'll start with you if that's okay. And I think that your slides will start right now. You could just speak to them and ask them to be advanced.
Fantastic. Thank you so much. Appreciate it. So I'll just ask to advance slides or animation as we progress. So I debated on how to introduce you all to Olink if you haven't heard of this growing Swedish-based company that came out of Uppsala University. I decided to use this slide. It shows an early version of Olink's protein library. It's based upon abundance in plasma for convenience. So here we're talking proteomics. The scales off to the right from above milligram per mil to below picogram per mil. So the highest abundant proteins are in the upper left, and they're well characterized with mass spec methods. Can we advance the animation? Great. So, those high abundant proteins, many of those have been moved to the clinic. Examples I use in this slide are HDL and LDL. What's been challenging is what we're highlighting here, which is the signal from proteins in the lower right quadrant. And Olink concentrated on the lowest abundant proteins to complement existing methods. And you can see the density of the dots here. So researchers have generated over 1,000 peer-reviewed publications by being able to look at a broader view of the proteome. Today in the meeting, we're talking about potential industry partnerships and the value of longitudinal cohorts. I spent 14 years at Illumina steeped in genetics. I think proteins are coming of age now and are helping us understand more real-time health in the context of genetic information. Next slide. And you can advance the animation on this as well, thanks. So the foundation of the technology is what's called the proximity extension assay, or PEA. Here the Olink Royal We, the R&D group, developed 2 antibodies per protein, and these are conjugated to single-stranded oligos so that they can hook out proteins, and we can work in 6 microliters of fluid to hook out these low abundant proteins, the broad proteomic look. And then those oligos can hybridize, and now you've got essentially a library prep that can be read out on a qPCR or a DNA or an NGS instrument. So currently we can do about 3,000 proteins for 384 samples. We had doubled our library from 2020 when it was launched to today. It's around 3,000, and we plan to expand further in 2023. So how are people using our technology? Next slide, please. Great. So this is just highlighting a project that came on the heels of the Exome Sequencing Consortium. It's called the UK Biobank Pharma Proteomics Project. And 10 pharma partners contributed to this to do over 50,000 samples of UK Biobank participants. Next slide. Today, this has expanded to 13 pharma partners with the addition of Novo Nordisk, Calico, and Alnylam, who joined in once Olink was selected for the proteomics platform. And we have 3 additional industry partners that are interested in potentially a next phase, which I hope might include more diverse cohorts. Next slide. And this is highlighting the first preprint from the UK Biobank Pharma Proteomics Project. This posted in June of this year. The analysis team, this is led by Ben Sun, identified over 10,000 pQTLs in 54,000 individuals. Over 85% of these they reported as novel. And I would say, imagine discoveries in cohorts with more diverse ancestry. These are exciting times, I think. Next slide and last slide, and you can advance the animation on this one as well. Great. So we believe a biomarker should provide not just a biomarker, but it should also provide mechanistic biology and potentially a druggable target or a potential therapeutic target information. I'm excited about how genomics and proteomics and other omics like metabolomics will drive a more systematic approach to therapeutic target identification, and essentially precision medicine. Thank you.
All right. My name is Peter Wirtz. I'm Scientific Director for Nightingale Health, and I will be presenting here how our metabolomics platform is enabling full cohort profiling to pioneer and push the limits of precision medicine. So the unique features of Nightingale's biomarker profiling platform is the fast pace and affordable profiling. We can use a standard blood sample, 100 microliter, is profiled using a high throughput NMR platform that in large sample numbers enables a very cost-effective biomarker profiling. And we worked successfully with IHCC cohorts and continue to offer an attractive price point at around 20 euros per samples. So what do you get from the biomarker profiling? What you really get is biomarkers that are relevant for future disease risk. A broad metabolic profile, including lipid measures, apolipoproteins, fatty acids, and a range of small molecules like amino acids and ketone bodies. And together they cover different metabolic pathways like inflammation, fluid balance, glycolysis. And that combination of different metabolic pathways in a single measurement really allows quite substantial disease prediction, much better than individual biomarkers when you combine the biomarkers into scores. And I would like to highlight a recent paper in Nature Medicine on prediction of multi-disease outcomes, where 24 common disease outcomes was aimed to predict with this single metabolomics assay, and showing very promising results in terms of improved prediction. And if you're more interested in the individual biomarkers, I would like to highlight this other paper, Atlas of Biomarkers versus Health and Disease, in the first results of UK Biobank participants with over 100,000 individuals and results across over 700 disease outcomes. In addition to these emerging, exciting, novel biomarkers, I would also like to highlight that Nightingale panel actually includes many clinical chemistry measures, routine biomarkers like apolipoproteins, LDL and HDL cholesterol, creatinine, glucose, and more emerging biomarkers like omega-3 and omega-6 that are very well understood and relevant for health and disease. And in total, 39 clinically validated biomarkers with CE mark. And what that really means is that you get basically the same results of the biomarker measures from Nightingale as the standard clinical chemistry. And that allows you to replace many of the standard lab tests. And also these routine biomarkers enable to provide, for instance, the exciting results of genomic prediction more into clinical context. So this combination of fast, affordable profiling and a broad metabolic profile has made Nightingale platform quite popular in many of the largest biobanks in the world. Like we're profiling all the samples from UK Biobank and working with many other cohorts in the IHCC consortium, and are really excited to scale this out to many broad diverse ancestries. The results from the different cohorts replicate really quite nicely, and we heard yesterday Hakan and co-workers telling about successful pilots in the IHCC consortium. So by profiling many cohorts and replicating and validating results, we can use this consortium to really push the limits of precision medicine and really move beyond biomarker discovery to improved prevention. So I could talk for hours about the scientific opportunities, but if you want to hear more about how this profiling could benefit your cohort, then tune into this IHCC webinar coming up in a few weeks' time. And meanwhile, let's be in touch and discuss how this high throughput metabolic profiling can benefit the scientific aims of your cohort. Thank you.
Scott, you're on mute.
Sorry, Heiko. Would you care to go next, please?
Okay, I have not prepared slides, but I hopefully can speak to Biogen, their interest in international cohorts, and my particular role in this. I'm Heiko Runz. I'm leading the human genetics group at Biogen, which is not a technology-focused company as the other two speakers represent, but rather a biopharmaceutical company who's applying those technologies to large-scale population cohorts up to individuals participating in our clinical trials, and who are taking interpretations from those data sets to inform our drug discovery and development programs. The entire paradigm is based on the idea that drug targets that are supported by genetics and human data have a higher probability to succeed in clinical trials and make it to actually successful medications. Biogen's focus is on neurodegenerative and neurological diseases. We have marketed products for multiple sclerosis, for spinal muscular atrophy. We have a very strong research pipeline for a range of neurodegenerative diseases, and we are highly interested in ascertaining information both from individuals with those diseases, as well as more and more individuals that are either healthy controls or still healthy and pre-symptomatic, but will develop those diseases in the future. We are using these data for making predictions whether interventions against a distinct drug target will likely be efficacious, whether we can expect safety signals when we are developing drugs that are phenocoping what we will identify from those human data sets. And for example, to identify new biomarkers that may predict disease progression or the success of interventions that we are conducting against a particular disease. What is more and more relevant for us is also to identify individuals with subtypes of diseases, for example, to develop clinical trial protocols with the highest probability of success. So also just to identify participants for our clinical trials or future customers for our drugs. And more relevant for this is also real-world health information that is being captured in many biobanks that we're utilizing, not only for post-marketing questions, but more and more also in research and development to define the disease indications, better understand the diseases that we are aiming to treat, and also better understand epidemiology or geographic distribution. So I'm really excited to be on this panel today and with so many international participants.
Thank you, Heiko. Okay, we can turn to some questions that I've prepared here, and we also will be able to take questions from viewers. You can put those in the Q&A on the Huba platform there for this session. But let me start, and I'll start, Heiko, with you, just because I think you may have embedded the part of this in your overview. But what do you see as the value of IHCC, its cohorts, and its membership, particularly to Biogen as a biopharmaceutical company?
So very happy to speak to this. In fact, if you look back in 2018, I believe this is when I joined the company, we had hardly any of our drug discovery programs based on genetic data or thorough large-scale population data. Often our hypotheses had been drawn based from small data sets or animal studies, and human data sets as they are being captured in these large-scale biobanks have really grown substantially over those last couple of years, so that now really the vast majority, both of our clinical pipeline as well as the preclinical pipeline, are based on either genetic hypotheses or human data sets that we have obtained in such clinical or population registry information. And we are using information on that really across all stages of our drug discovery and development programs. And one observation that we have made that it's typically, as we've learned from genetics, the larger the cohorts that we are working with, the more diverse the cohorts that we're working is, the more accurate are the hypotheses that we are drawing from them. So I think just the sheer diversity by having so many cohorts under one umbrella organization is of an enormous interest. And we are really, one of our key questions often is, what is the epidemiology? What is the global distribution of certain disease indications that we're going after, genetic variants that we are particularly interested in, and here as well, this vast diversity of a cohort like IHCC is very, very attractive and useful for our daily work.
Thank you, Heiko. Cindy, same question for you, the value of IHCC for Olink.
Sure. Yeah, it's a appreciated question. So, I mentioned in my intro the importance of understanding diverse cohorts and not lagging behind on understanding the proteome the way we have in the genetic space. Although, we have reasons for why we're in this position, and we're trying to advance a genetic understanding of a more unbiased view of ancestral diversity, as well as an unbiased view of the proteome in general. So I guess I think about the IHCC as a highly incentivized group of cohort leadership that helps define what's needed to move us to precision medicine. From Olink's perspective, we're a platform company, unlike Heiko's representation of Biogen, who I think of as a user of our platform, as well as a definer of how we need to innovate in the future. And so I see this as a collaborative partnership of industry partners with cohort members who are trying to meet stakeholders' needs that Heiko just articulated so beautifully, and then where we need to innovate to keep up with that, to provide a more unbiased view of the proteome, as we've been able to see done in the genome. Right, we went from targeted genomics to a more unbiased, sequencing view of the genome. I see that as needed for a rising tide to lift all boats across ancestral backgrounds and advancing precision medicine for all ancestries. Hopefully that made sense.
Oh, thanks, Cindy. It does. It does. Peter, lastly, how about you? What value do you see in IHCC, its cohorts, and its members?
Yeah, thank you. So I think we have, as a company and the academic scientists, very similar aims in terms of getting the best possible science done and advancing precision medicine. It is very much in the interest of Nightingale, and I'm sure it's in interest of the individual cohorts and the consortium as a whole. And kind of IHCC and big cohorts are really enabling that in a way that the smaller scale individual cohorts are not able to. So we are really pleased to kind of work with these very large studies that can kind of leap the evidence instead of having it in small steps. And I think sometimes the biomarker discovery field has been a bit plagued with kind of that there's kind of reported a biomarker here and there in one study that is not holding up in another study. And I think really these efforts from the consortium to kind of combine the analysis across cohorts is really really interesting and important, essential for the field.
Thanks. My next question, I'm not going to ask Heiko this because I think you already answered it in that Biogen is a neuroscience company and that's their research interest. But Peter, let me follow up with you. Does the metabolomic approach, is that widely useful for research in particular disease areas, or is it restricted to metabolic disease, or if you could answer that? And I realize you're giving a webinar, and thank you for reminding me and everyone else that's listening about that in the middle of December, but could you speak to that?
Yeah, thanks. It's almost like a setup question. We started out with the focus on diabetes and heart disease, and particular emphasis on the detailed lipid profiling. But through the profiling of these large cohorts really has been the exciting findings most exciting findings from beyond cardiometabolic diseases like liver diseases, lung diseases. Certain cancers have shown that the biomarkers are relevant, and even in like neurological diseases, including dementia and infectious diseases like susceptibility to severe infections have been shown that these biomarkers are relevant. So I think the big cohorts and the science now coming out really changes how we understand that these biomarkers are relevant for more holistic aspects of medicine than we previously thought.
Cindy, same question for you. Olink's technology widely applicable?
Yeah, sure. So I just also wanted to mention something about what Peter is talking about. I do also find that metabolomics and this system-wide approach gives such insight into microbiome and its influence on metabolic disease and other diseases. In terms and the layering of proteomics onto that, I think is a very exciting aspect. I said, and I use this analogy quite a bit, that I think of this as a rising tide that lifts all boats. I think of that more like as a disease agnostic approach, as you hint at, Scott. But areas where groups have made advances, particularly in being able to pull out these low abundant protein signatures or multi-analyte signatures, include consortia. We have the Coral Consortium on neurological disease, proteogenomic efforts that are more like this disease agnostic approach, like SCALLOP, like the UK Biobank Pharma Proteomics Project, but also advances in cardiovascular disease, a dozen different signatures in things like ovarian cancer, multiple sclerosis, autoimmune diseases as well. So I think it's quite broad, but that said, there are pockets of advances that I'm excited to see. And then I've had lots of inquiries and questions about NASH and NAFLD cohorts. So lots of growing interest there.
Thank you. Okay, I'm going to skip Peter on this question because he already talked to us about how Nightingale is collaborating with IHCC. But Heiko, how could you envision a collaboration between IHCC and Biogen evolving?
So I think, speaking particular for Biogen, we have made very good experiences with pre-competitive partnerships where in a way we are co-developing resources or cohorts together. So that means industry partners, not just one single industry partners, but industry partners often in a team are teaming up with academic researchers or individuals who are generating data on those cohorts to well-define research questions, identify what are opportunities for data generation, and then draft a work plan, decide what are the most appropriate tools and technologies to use, and then go for a data sharing model where very often the data that is being generated itself is considered of relatively modest intellectual property risk, but it's typically made broadly available because we do want to share out information like this for enabling broader research. So overall, our collaboration model is fairly open for the large majority of data, and we are highly interested in, say, federated data models where data is being generated in individual cohorts and then being brought together in a central resource. And I guess some of my dreams would be that I can just send an email out and ask around, provide me information on how many individuals do you have with these and these characteristic, genetic, clinical, biomarker criteria, and what are the data sets that I would like to, or the cohorts that I would need to go into for further in-depth follow-up studies.
Okay, thank you. So I'm going to go to a question from the audience. Jeff Ginsburg asks, is there a need for a reference proteome or metabolome with diverse representation from different ancestral backgrounds, or is that just too great a challenge given the influence of environment, et cetera, on these types of analysis? And Peter, let's start first with you on that.
Yeah, I think the most analysis on different ancestries are showing that it's fairly similar biomarker disease results, at least in terms of association magnitude. But I think there's a huge value that IHCC can provide with kind of profiling this more broadly. I think the vast majority of cohorts we have profiled are from Europe. So there's a lot of opportunities to discover this further. One potential challenge to highlight is if the sample collection protocols are slightly different, then that may give subtle differences in the concentration values that can be different environmental factors, genetic background, but can also arise due to how the samples have been collected or stored.
Mm-hmm. Mm-hmm. Thank you. That's very, very thoughtful. Cindy, how about you? A reference metabolome or a proteome?
Yeah, great question. So this is, top of mind, that graph I showed with 1 dot per protein. You can imagine for every one of those proteins, there's a whisker that represents the dynamic range in a healthy sample, but also in a disease sample. You want to capture that in any given assay. And so challenging these assays against different conditions are essential. And same thing around ancestry, right? So characterized primarily today are Northern European populations, and we have a free interactive tool for pathway browsing where you can see the reported proteome it's just around 500 samples of Northern European ancestry. Those are going to be quite different for different ancestral backgrounds, right? Because you can think about how proteins are encoded by the genome. We know the genome is going to be different. So those exposures over a lifetime to different protein levels might be protective or might be increasing risk for disease. And those discoveries are still yet to be made, right? And then I'll just say one more thing is, as you bolt on additional, we've definitely seen in modeling studies, as you bolt on additional samples onto existing data, you continue to make these protein quantitative trait loci associations, those discoveries, at least to the 35,000 samples we've modeled today. Those represent pathway relationships that may be linked to disease or might be in common between different diseases. And I think understanding that mechanistic biology in the context of different ancestral backgrounds is going to tell us a lot about polygenic risk, polyprotein risk, and how to better improve, like Peter said, precision medicine.
Okay, last question. One of IHCC's major missions is to bring genomic science to low to middle income countries and low resource settings. Is there particular activities that your organizations would be interested in participating in that would help that objective? And I'll start first with you, Heiko, on this one. Is there something that would be centered on LMICs that would be of value to Biogen?
Oh yes, definitely. So I mean, our entire population level research has so much centered on Europeans in the past decades. It's really the time that non-European populations are much more represented in research, and they provide substantial additional information. We know that already at substantial lower sample sizes, just because of sheer differences for genetic data sets, for example, variant population frequencies, we often have a substantially different association or power for us to detect new disease associations. And often this can be achieved at much smaller sample sizes. So for certain diseases, indications, non-European cohorts are of substantial value. I guess what we are also more and more interested in, particular for our drug discovery efforts, which are typically addressing global populations, is that we have more representation and just in our clinical trials. And for that, we do need to have better recruitment mechanisms than are currently happening for our clinical trials. So just having the option to go into these non-European population cohorts and get an overview of what individuals, what information may be available on participants, and how to eventually even establish ways to recontact those individuals or obtain samples from trial participants or from cohort participants is of a substantial interest. So I very much encourage also younger researchers to really develop their research programs to being to really systematically ascertain such data sets, as well as the infrastructure to leverage these cohorts.
Thanks, Heiko. The organizers are telling me that we are out of time, so I'm going to have to save that question till later. But I want to thank you all for being on this panel today. As I guessed at the beginning, we could have spoke about many questions for a much longer time. That means that I might have to ask you back for a repeat performance sometime in the future. I hope you'll all agree. And thank you very much for participating today. We truly appreciate it.
Thanks, Scott. And
thank you very much.
Organizers, amazing team.
Let me just orient people. There is a short poll that's going to be coming up in a second as part of our icebreakers. I believe this one is called 2 Truths and a Lie, and it will be mostly around IHCC facts, so you won't have to reveal anything personal. And then that will be followed by 2 roundtables that'll be coming up. You have your choice between 2 of them. One is on the Policy Starter Pack, and then the other one is on trans-ethnic and analytical methods. You can choose between either one of those. Once you've chosen one, you can always switch to the other one. You're not stuck, but you will need to go to those sessions and select one of those 2 roundtables after the short icebreaker. And thank you again, panelists, for all that you've done. Bye now.